The in situ proximity ligation assay, or PLA, is a technology capable of detecting interactions among proteins in fixed tissue and cells. The associations between proteins can be identified and quantified without the need for transgene expression or additional text. The only requirement is the availability of specific high-affinity antibodies that can be modified with DNA oligonucleotides. In tissues, MIST1 and MIST2 exist mainly as active homodimers, but oncogenic stimuli can increase level of MIST1, MIST2 heterodimers, and such heterodimers are inactive. Following incubation with specific primary antibodies against the proteins of interest, species-specific secondary antibodies, each linked to a unique short complementary DNA strand attached to it, are added to the slide to bind to the primary antibodies. If the proteins in question are heterodimerized, both the primary antibodies and secondary antibodies bound to them will be in close proximity. In this state, the attached DNA strands on the secondary antibodies can interact through a subsequent addition of two other circle-forming DNA oligonucleotides. Several hundred-fold replication of the DNA circle can occur after an amplification reaction, and a fluorescence signal is generated by labeled complementary oligonucleotide probes. Therefore, each detected signal is visualized as an individual fluorescent dot, which can be assigned to a specific subcellular location based on microscope images. On the day before experiment, coat 16 wheel chamber slides by adding 50 to 100 microliters of mouse laminin or poly-L lysine and incubate at 37 degrees Celsius.